Good day, everybody. My name is Katy Leung. I'm from Singapore General Hospital and Duke NUS Medical School. Today, I'm going to talk about the future of biomarkers for psoriatic arthritis. So a biomarker is a measurable indicator of severity or the presence of a disease. So it can be classified in a number of ways. If you talk about imaging, it would be imaging biomarkers like X-ray, MRI, and ultrasound. There could be clinical biomarkers, which include physical sign, a physical status that a patient in. Of course, we can talk about the biological process that you measure it in blood, urine, synovial fluid, or even tissue. Of course, the other way of classifying it is according to the function of the biomarkers. It's like whether it is a biomarker of a presence or absence of disease is a diagnostic biomarkers. We also have a biomarker of disease severity. We talk about differentiating severe disease from the milder disease. Of course, we also have biomarkers that predict drug response or interventional response. So out of all these functions, I think the most interesting will be biomarkers that predict the treatment response. So as we know that up to 30% of our psoriatic arthritis patients, they do not have adequate response even to the most powerful biologic or synthetic DMARDs. So if we shift them into another class of drug, interestingly, they would have some kind of response. So, but we cannot predict what the response of a patient towards drugs. So what we have been using now is looking into the domain that this patient is affected and also looking into comorbidity and make a best guess. But however, a lot of time is that the best guess doesn't work. And then we have to shift again after 3 months or half a year, which means there is a waste of time and also a waste of opportunity. So how wonderful it might be if there are some biomarkers that help us to predict response. There are some of these biomarkers has been studied, including the CRP, the plasma C3 level, interleukin 16. There are a study using the global review trial data set and looking into a 14 biomarkers panel and try to predict a response. And some of these biomarkers, including interleukin 16, the VEGF, the MMP3, and CRP. These things have been proved to be more useful than CRP alone. As for tissue biomarker, which some group of researchers have studied in rheumatoid arthritis, have been employed in psoriatic arthritis. So they take tissue, synovial tissue from patients, and try to use these to predict a DES28 response. And some of these biomarkers in a combination is proved to be quite useful. However, all of these studies need more validation. Of course, the most interesting part will be a study coming from the Japanese group. They try to use a cytometry method to distinguish between an interleukin 1 signal and interleukin 17 signal. So from this interleukin 1 signal and interleukin 17 signal, they have 4 groups and stratify accordingly and give them the appropriate treatment in the stratified group as compared to the standard group. Interestingly, what they found is that the stratified treatment group has almost double the clinical response as compared to the standard treatment group. Although this is a very tiny study that will need a lot of validation in future, it is indeed a proof of concept that we can use biomarkers to predict treatment response and help our patient to better choose the treatment of choice. Of course, needless to say that although we have all these biomarkers, it is still in its infancy and needs a lot of modification and validation. Some of the challenges include that the variability in the standardized protocol. there's lack of a standardized operation procedure in all of the things, including the sample collection, in processing, in transport, in storage, and also the experiment and analytical analysis. So, of course, we also have other obstacles like the biological diversity, age, sex, ethnicity, lifestyle, diet, etc. And of course, we also have the disease heterogeneity for psoriatic arthritis. It might be the peripheral joint arthritis, it might be the psoriasis, might be the enthesitis or dactylitis. Each of these will affect how we define outcome. What is responder as compared to non-responders? But a group of international collaborators, as collated by the GRAPPA, has been putting all these things together, as led by Professor Chris Richland and also Oliver Fitzgerald. They are putting a collaboration of researchers. We call them CRN. Then they are developing this kind of standardized protocol. So majority of the biomarker studies are coming from single center, and the sample size is still small. Majority of them are looking at one modality. Of course, now we have advances in technology looking into the omics that can help us to deeper understand the pathogenesis of psoriatic arthritis. We have several kinds of omics technology, like transcriptomes, proteomics, and immunomics. Transcriptomes mean that we are having a snapshot of what the cells are making in the RNA. It's a snapshot of all the RNA the cells are making at the time of sampling of the sample, whether it is a skin sample, blood samples, or synovial fluid sample, and even tissue samples. So it's a snapshot. And proteomics is at the time what the cells are making, all the proteins. These are the effector protein that is making the cell functioning. And immunomics is talking about the cell type, no matter it's T cell, it's B cell, and all of these are going into very deep understanding, going to the level of single cells that you can look into the different cell type of T cells, you can look into different cell type of B cell. One of these key cells that is very interesting is the Treg cells. They are making a lot of impact over there. So I would be staying optimistic about biomarkers because the advancement of technology is there. We have different portals, and in future we can combine all these portals to give us a better understanding of pathogenesis and also help us to evaluate for the biomarkers. We have the unmet need is there, it's huge. We don't know how to predict treatment response. We have the science and we have the technology, and we can combine modalities. We have standardized protocol and procedure in the future, and I'm sure we will be able to find something for the future to make our patients better. Thank you for your attention.